For almost all of human history, living to 40 years old was not normal. It was rare. In prehistoric times, many lives ended during childhood, during childbirth, or from injuries and infections that today feel minor. The human body was built in small groups, moving through dangerous places where surviving long enough to have children was good enough for nature. Natural selection does not promise long decades of getting old. It keeps what works right now. If most humans in prehistory never reached 40, why was that the case? And what does it tell us about the kind of life our bodies were originally built to live? For most of human history, the world that shaped our species was unstable, huge, and did not care about any one person's survival. Modern humans first appeared in Africa about 300,000 years ago during the Pleistocene period, a time marked by repeated cycles of ice ages coming and going. These climate changes did not just change the temperature, they reshaped land, shifted animal movements, changed which plants were available, and forced small human groups to keep moving in search of food and water. In such a world, daily life was focused on surviving right now rather than planning for the future. When we ask why most humans in prehistory never reached 40, we must first understand the pressures from nature that shaped every stage of life from birth onward. Early human communities were small, often just a few dozen people, and moving around was necessary. Staying in one place too long risked using up all the food nearby. Moving required energy, exposed people to unfamiliar land, and increased the chances of running into predators or rival groups. The environment offered no steady supply of food. Food came and went with the seasons, droughts, and animal patterns. Times of plenty could quickly become times of starvation. Under these conditions, just surviving to adulthood was already a big achievement shaped by luck and toughness. The human body evolved within this pattern of uncertainty. Natural selection favored traits that made it more likely to survive long enough to have children, not traits that made sure people lived comfortably for decades afterward. The way the body uses energy reflects this. Growing during childhood, starting puberty, and being able to reproduce were the most important biological goals. Long-term tissue repair, protection against age-related breakdown, and the slow maintenance of cells beyond middle age were not strongly pushed by evolution if most people did not consistently live to old age. In the Pleistocene landscapes where Homo sapiens first spread beyond Africa into Eurasia, dangers were real and close. Large predators like lions, hyenas, and ice age hunters shared the same territory. Getting injured while hunting large animals could be very bad. Even small wounds risked infection without any way to clean or treat them. Exposure to cold climates during ice ages added more strain. Archaeological evidence from early sites shows healed broken bones and joint damage, which tells us that some people survived serious injuries, probably because others in the group helped them. But those same skeletons also show injuries that likely ended many lives too early. The environment did not need to be catastrophic to shorten a life. It only needed to be consistently hard. Infant survival was especially fragile in these settings. High rates of childhood deaths lower the average age at death in any group, even if some adults lived into their 50s or beyond. When researchers estimate life expectancy in prehistoric groups, they often find averages below 40 years. This does not mean the human body was unable to reach that age. It means that the combination of environmental dangers, infectious disease, poor nutrition, and no medical care made it statistically uncommon. The evolutionary context is very important. Our ancestors did not evolve in settled cities with stored grain and clean water. They evolved in shifting ecosystems where survival depended on working together, adapting, and physical endurance. The pressures that shaped the human genome were focused on early and middle life. The later decades that many people experience today were rarely stable or predictable in deep prehistory. Understanding this ecological background allows us to approach the main question with more clarity. If most humans in prehistory did not reach 40, the answer is not found in one single cause, but in the heavy weight of an environment that demanded constant toughness from bodies built for immediate survival rather than long life. The instability of the prehistoric environment did not only shape where humans moved, it shaped how their bodies divided energy across a lifetime. In small hunter gatherer groups, survival depended on a careful balance between effort and reward. Calories gotten from gathering or hunting had to cover immediate energy needs, support growth in children, sustain pregnancy and breastfeeding, and fuel constant physical activity. There was little room for long term biological investment beyond what was needed for reproduction and short term survival. When thinking about why most humans in prehistory never reached 40, it becomes important to understand how energy limits quietly shaped the aging process itself. The human body is not a machine built for endless repair. Cell maintenance requires resources, repairing damaged DNA, replacing worn tissues, supporting immune defense, and keeping organs working all use energy. In an environment where food availability went up and down and periods of scarcity were common, the body focused energy on surviving in the present. Reproduction, immune response to immediate threats, and quick healing of serious injuries were the priorities. Slow damage that might show up decades later did not strongly affect the ability to have children in a population where many people did not consistently live too old. Age, this pattern matches what evolutionary biologists call trade-offs. A trait that improves early survival or reproductive output can stick around even if it causes problems later in life. In the ancestral environments of Africa and later Eurasia, early adulthood represented the peak of reproductive value. The biological systems that support muscle strength, fertility, and handling stress were strongly maintained. Systems that guard against gradual breakdown beyond middle age were under weaker selective pressure over thousands of generations. This balance shaped a body built for resilience under immediate stress rather than long-term stability into old age. Climate changes during the Pleistocene made these pressures stronger. Rapid shifts between warmer and colder phases altered vegetation and animal distribution. Archaeological records show that human groups adapted through tool invention, social cooperation, and flexible diets. Yet adaptation did not eliminate risk. Nutritional stress left marks on skeletal remains in the form of enamel hypoplasia in teeth, showing periods of childhood malnutrition. Repeated stress during early development can have long-term physical consequences, affecting immune function and metabolism. Such early life hardships increase vulnerability to disease and may shorten life even if a person survives into adulthood. The structure of prehistoric populations further reinforced this pattern. With high rates of infant and juvenile deaths, the age distribution was heavily weighted toward the young. In a community where many members were children or young adults, relatively few people were in their later decades. This demographic structure influenced social roles, the passing down of knowledge, and work patterns. Elders did exist, and studies of some modern hunter-gatherer societies show that individuals can live into their 60s or beyond under similar ways of living. However, the proportion of such individuals remains smaller than in modern industrial populations. The environment did not reliably support large numbers of people reaching old age when researchers estimate average life expectancy for prehistoric populations. They rely on analyzing skeletons and demographic modeling. These methods suggest that life expectancy at birth often fell below 40 years, largely due to early deaths. If an individual survived childhood and the teenage years, their expected remaining years increased, but even then, persistent environmental hazards limited how long they lived. The body's biological design must be understood within this demographic and ecological framework. It was shaped in conditions where surviving long enough to reproduce was sufficient for genes to continue. Extended post-reproductive lifespan, though possible, was not the statistical norm in many prehistoric settings. The combined effect of energy constraints, environmental instability, developmental stress, and demographic structure created a world in which reaching 40 was less common than it is today. This does not imply that humans were incapable of living longer. It suggests that the environment in which our species evolved did not consistently allow most individuals to do so. Beyond climate and energy limits, another quiet force shaped the lifespan of prehistoric humans. Constant exposure to physical risk in small mobile groups moving across open savannas, forests, river valleys, and later colder steppe landscapes. Daily survival required direct engagement with the environment. Hunting large animals demanded close contact with powerful prey. Gathering exposed people to unfamiliar terrain, hidden hazards, and venomous creatures. Even routine movement over uneven ground carried the possibility of falls or broken bones. In a world without surgical care, antibiotics, or controlled sanitation, the line between a survivable injury and a fatal complication was narrow. When asking why most humans in prehistory never reached 40, this persistent background of risk cannot be separated from the ecological picture. Archaeological evidence quietly speaks to these dangers. Skeletal remains from various Pleistocene sites show healed broken bones in ribs, limbs, and skulls, suggesting that injured individuals sometimes survived long enough for bones to heal. This implies social cooperation and caregiving within early human groups. At the same time, many skeletons show trauma that likely contributed to early death. Embedded projectile points, signs of head injury, and degenerative joint damage reveal lives shaped by physical strain. Even when healing occurred, repeated injuries would pile up over time, placing stress on joints and reducing mobility well before what modern societies consider old age. The body could endure hardship, but its resilience had limits. Infectious disease added to these risks. Bacteria do not need dense cities to spread. They thrive wherever humans gather, share food, and interact closely. Minor cuts could become infected. Gastrointestinal illnesses from contaminated water or food could lead to dehydration. Parasitic infections were common in many environments and may have caused chronic weakness. While some modern hunter-gatherer societies show relatively low rates of certain epidemic diseases compared to agricultural populations, localized infections and untreated wounds would still have taken lives. Without antibiotics or advanced medical help, the immune system bore the full burden of defense. If it succeeded, survival continued. If it failed, life ended, sometimes quickly. Childbirth presented additional danger. For women, reproductive years carried repeated physical stress. Pregnancy and breastfeeding required substantial energy, and complications such as obstructed labor, heavy bleeding, or infection could be fatal. Skeletal and ethnographic evidence suggests that maternal mortality in pre-modern contexts was significantly higher than in modern industrial societies. Each pregnancy was both a biological necessity for population survival and a period of elevated risk. Over time, repeated reproductive stress could weaken the body, making survival into later decades less certain. These layers of risk interacted with demographic structure. In a population where infant and juvenile deaths were already high, the loss of adults in their 20s or 30 seconds further reduced the proportion of individuals who might approach 40 average. Life expectancy remained low not because no one could live longer but because cumulative hazards across the lifespan limited how many did. When researchers model prehistoric populations, they often find that surviving to the teenage years significantly improved an individual's chance of reaching later adulthood. Yet even then, the environment continued to present threats that modern societies have largely reduced. Returning to the origins of our species in Africa and its later expansion across diverse ecosystems, it becomes clear that human biology was formed in this context of layered vulnerability. The genome reflects selection for flexibility, cooperation, and short-term resilience. It does not contain guarantees against infection, injury, or reproductive risk. Longevity beyond 40 was possible and did occur, but it unfolded against a backdrop where daily life carried real danger. Understanding this cumulative exposure to risk helps clarify why reaching 40 was less common in prehistory. It was not a fixed biological ceiling. It was the statistical outcome of living in environments where the margin for survival remained thin across every stage of life. When researchers reconstruct the age structure of prehistoric populations, one pattern appears consistently across regions and time periods. Childhood was the most fragile stage of life. In many ancient communities, a significant proportion of individuals died before reaching the teenage years. This fact alone reshapes how we interpret the idea that most humans in prehistory never reached 40. The average age at death in a population is heavily influenced by infant and juvenile deaths. If many children die young, the overall life expectancy at birth drops sharply, even if some adults survive into their 50s or beyond. Understanding this demographic structure is essential for approaching the main question with clarity rather than assumptions. In small hunter-gatherer groups during the Pleistocene, childbirth happened without surgical intervention, sterile environments, or advanced monitoring. Newborns faced immediate vulnerability. Premature birth, low birth weight, birth defects, and infection could all threaten survival in the first days or weeks of life. Breastfeeding provided immune protection, but the mother's health and nutrition influenced both milk production and the infant's resilience. In times of scarcity, infants were especially at risk. Skeletal remains from prehistoric sites often reveal a disproportionate number of subadult individuals, suggesting elevated deaths before adulthood. While preservation biases must be considered, the demographic modeling of such populations consistently indicates high early life mortality rates. This early vulnerability connects directly to our species' evolutionary beginnings. Homo sapiens evolved a long childhood compared to many other mammals. Extended juvenile development supports brain growth and social learning, which are defining features of our lineage. Yet a prolonged dependency period also increases exposure to risk. Children rely on adults for food, protection, and care. If adults are injured or resources decline, children suffer first. In unstable ecological conditions, such as those of ice age cycles and fluctuating climates, the survival of young individuals becomes uncertain. A demographic pyramid heavily weighted toward the young reflects both high fertility and high mortality. From a statistical perspective, this structure lowers average life expectancy without requiring a biological limit at 40. If half of a population dies before 15, the average age at death may appear very low, even though survivors past the teenage years may live decades longer. Researchers often distinguish between life expectancy at birth and life expectancy conditional on surviving childhood. For individuals who reach their teenage years, the probability of living into their 30s or 40s increased significantly. Nevertheless, cumulative environmental hazards continued to shape outcomes. The key point is not that human biology prevented anyone from reaching 40, rather, the combination of high early mortality and persistent adult risk made it uncommon for most individuals within a population to do so. Ethnographic studies of some contemporary hunter-gatherer societies provide cautious comparative insight. In groups with minimal access to modern medicine, child mortality remains higher than in industrial nations, and life expectancy at birth can fall below 40, yet adults who survive the early years often live into their 50s or 60s. This pattern likely reflects aspects of prehistoric demographic structure, though direct comparison must remain careful. Environmental conditions, contact with outside populations, and ecological context differ. Still, the general principle holds. High early mortality strongly shapes population averages. When we return to the prehistoric record, it becomes clear that the phrase never reached 40 does not imply an intrinsic biological cutoff. It reflects a demographic reality shaped by fragile childhood survival. The evolutionary environment that forged our species did not guarantee safe infancy or stable adolescence. Many lives ended before full adulthood, pulling the population average downward. To understand why 40 was less commonly attained, we must first recognize that the earliest years of life carried the greatest uncertainty. In that uncertainty lies a large part of the answer. To understand how strongly early mortality shaped prehistoric lifespan. It helps to imagine the age structure of a small mobile group tens of thousands of years ago. In such a group, children and adolescents likely made up a large proportion of the living members. Birth rates were relatively high in order to offset frequent loss. Yet each birth carried uncertainty, and not every child survived the vulnerable early years. When demographers model populations with high infant and juvenile mortality, the resulting average age at death falls dramatically, even if those who pass the teenage years may live for decades. This statistical reality lies at the heart of why most humans in prehistory did not reach 40. It was not that bodies were programmed to stop at a fixed age. It was that many lives ended long before adulthood was complete. In prehistoric environments shaped by climate changes and resource variability, childhood survival depended on multiple fragile conditions all working at once. A mother needed enough nutrition to carry a pregnancy to term and produce adequate milk. The group needed access to consistent food sources. Clean water had to be available, though clean in pre-modern contexts rarely meant free from microbial contamination. Exposure to pathogens occurred through soil, animals, and close human contact. Children's immune systems, still developing, faced repeated challenges. Some infections were survived and left immunity behind. Others proved fatal. The archaeological record offers indirect evidence of these pressures. Skeletal remains of subadult individuals frequently display signs of physiological stress. Linear enamel hypoplasia on teeth, for example, shows episodes during childhood when growth was temporarily disrupted, often due to malnutrition or illness. Such markers reveal that early life in prehistoric settings involved repeated stress events. Even when a child survived these episodes, cumulative physical strain could influence long-term health. Growth disruptions are associated in modern research with later vulnerability to metabolic and immune disorders. In prehistory, such compounded stress may have reduced resilience against future injury or disease, shortening lifespan further. Demographic structure also influenced social dynamics. In a population where many members were under 15, the passing down of knowledge relied on a smaller number of older adults. If elders died before reaching advanced age, generational overlap was limited. This does not mean there were no older individuals. Archaeological findings confirm that some humans survived into their 50s or beyond. However, their proportion within the population remained relatively small. When the majority of individuals are young, the cultural memory of the group compresses into fewer decades. This demographic compression reflects a life cycle shaped by early vulnerability rather than late. Biological shutdown.it is important to distinguish between maximum lifespan potential and typical lifespan realized under environmental constraint. Modern humans, even without advanced medical care, are biologically capable of living beyond 40. Contemporary small-scale societies without industrial healthcare demonstrate that adults who survive childhood can reach older ages. Yet the difference between potential and probability remains crucial. In prehistoric conditions, the probability of surviving early hazards was limited. Each loss in infancy or adolescence reduced the proportion of individuals who might eventually approach 40. When researchers calculate life expectancy at birth for prehistoric populations, the figure often appears strikingly low. This number reflects the combined weight of early deaths rather than a universal ceiling imposed by biology. Recognizing this helps prevent misunderstanding. The phrase never reached 40 describes a statistical pattern, not a rigid biological expiration date. High early mortality created a demographic landscape in which relatively few individuals accumulated enough years to enter what we now call middle age. In that landscape, the average lifespan was shaped less by the gradual processes of aging and more by the fragile threshold of early survival. The statistical weight of childhood mortality does more than lower an average. It reshapes how an entire society experiences time. In a prehistoric group where many individuals died before the teenage years, the visible face of the community would have been overwhelmingly young. Children learning to walk, adolescents acquiring skills, young adults entering reproductive years. These stages dominated the demographic landscape. The relative scarcity of older adults was not necessarily because aging itself was rapid, but because fewer individuals accumulated enough years to reach later decades. When asking why most humans in prehistory never reached 40, it becomes necessary to look not only at biological vulnerability, but at how repeated early loss structured generational turnover. In small scale hunter gatherer groups, fertility rates needed to remain high to sustain population size in the face of mortality. Women often experienced multiple pregnancies across their reproductive years. Each birth introduced risk. Yet without sustained fertility, the group could decline. High fertility combined with high early mortality creates a population pyramid with a wide base and narrow top. In such a structure, even if some adults survived into their 40s or 52nds, they would represent a minority within the living community. The majority would be clustered in younger age brackets. Over generations, this demographic pattern becomes normal. 40 would not mark a dramatic biological boundary, but it would mark an age few consistently reached. The archaeological record provides subtle confirmation of this structure. Burial sites from various prehistoric contexts often contain a high proportion of juvenile remains. While preservation conditions vary and may bias what survives, repeated findings of subadult skeletons across regions suggest that early death was a common part of life. Age estimation techniques based on dental eruption, bone fusion, and skeletal structure allow researchers to approximate age at death. These methods consistently indicate that many individuals did not survive into what modern societies consider middle age. The distribution of ages reflects both early mortality and continued environmental risk into adulthood. Generational overlap was therefore narrower than in contemporary populations. In modern societies, it is common for grandparents, parents, and children to coexist for decades. In prehistoric settings, such extended overlap would have been less frequent. This has implications not only for demographic averages but for social memory and cultural continuity. Knowledge transmission depended on fewer elders, and when older individuals did survive, their experience carried particular value. Ethnographic observations of modern hunter-gatherer societies show that elders often hold critical ecological knowledge, from migration patterns to medicinal plants. Yet the proportion of such elders remains smaller than in aging industrial populations. The limited number of older adults does not imply absence, but it reinforces the idea that long lifespan was not the dominant pattern. Returning to the origins of Homo sapiens in fluctuating Pleistocene environments, it becomes clear that demographic youthfulness was a structural feature of early human life. High childhood mortality compressed the age distribution, ensuring that most living members were young. Even if the biological potential to live beyond 40 existed, the probability of reaching that age remained constrained by cumulative hazards. The phrase "never reach 40" thus reflects a population-level pattern shaped by early life fragility and rapid generational turnover. It describes a world where time was measured less in decades of aging and more in cycles of birth, growth, reproduction, and frequent loss. Understanding this demographic rhythm deepens our view of prehistoric life and clarifies why 40, though biologically possible, was statistically uncommon. Reproduction stood at the center of prehistoric life, not as a philosophical idea but as a biological necessity. In small mobile groups spread across unstable landscapes, population survival depended on the continual birth of children. Yet each pregnancy and each birth carried measurable risk. When examining why most humans in prehistory never reach 40, the physical cost of reproduction, particularly for women, becomes a crucial element. The human body evolved to prioritize successful reproduction over extended longevity. This priority shaped not only fertility patterns but also the long-term durability of the body itself. Human childbirth is anatomically complex. The large brain of the human infant, a defining feature of our species, must pass through a pelvis shaped for upright walking. This evolutionary compromise between walking upright and having a big brain creates a narrow margin for safe delivery. In the absence of surgical intervention, obstructed labor could result in fatal outcomes for the mother, the child, or both. Hemorrhage, infection, and prolonged labor were common risks in pre-modern contexts. Skeletal and historical data from later pre-industrial societies indicate maternal mortality rates far higher than those observed in modern medical systems. While precise rates in deep prehistory cannot be measured directly, there is no evidence to suggest that childbirth was safer in Paleolithic environments than in early agricultural societies without medicine. Pregnancy itself required substantial physical investment. Nutrients consumed by the mother were redirected toward fetal development. In environments where food availability went up and down, repeated pregnancies could lead to chronic nutritional depletion. Iron deficiency, for example, increases vulnerability to infection and fatigue. Calcium demands influence bone health. In a context without dietary supplements or consistent surplus, mothers' bodies bore the cumulative cost of sustaining offspring. Breastfeeding extended this energy burden, often for years. High fertility in a high mortality population meant that many women experienced multiple pregnancies across their reproductive span, increasing cumulative physical strain. The evolutionary logic behind this pattern is stark. Natural selection operates most strongly on traits that influence reproductive success. If an individual survives long enough to produce and help raise offspring to independence, their genes continue. Survival decades beyond reproductive years exerts weaker influence on genetic transmission. This does not imply that older individuals lacked social value. Anthropological research suggests that post-reproductive adults can contribute to group survival through knowledge sharing, childcare support, and getting resources. However, from a strictly biological standpoint, the selective pressure to ensure survival beyond the primary reproductive window is less intense. In prehistoric demographic structures already shaped by high childhood mortality, reproductive age adults carried the burden of population continuity. Any mortality within this group had amplified demographic consequences. Yet the very act of reproduction increased exposure to risk. Women faced dangers in childbirth. Men often engaged in physically demanding hunting and territorial defense faced injury risk linked to providing for the group. The reproductive phase of life coincided with peak physical strain. Surviving through multiple reproductive cycles required resilience in environments that offered little margin for recovery. Archaeological findings from skeletal remains occasionally reveal pelvic injuries, consistent with childbirth stress. While direct attribution is complex, patterns of wear and trauma suggest that reproductive strain left lasting marks on the body. Even when mothers survived childbirth, cumulative stress could weaken resistance to later disease or injury. In populations without modern obstetric care, maternal mortality was a significant contributor to adult female mortality rates. This reality shaped the age distribution of communities, reducing the number of women who would eventually approach 40 when placed within the broader Pleistocene context in which Homo sapiens evolved. Reproduction emerges as both the engine of population survival and a source of biological vulnerability. The body was shaped to reproduce successfully under difficult conditions. It was not optimized for minimal wear across decades beyond that task. Understanding the reproductive burden clarifies one dimension of why 40 was statistically less common in prehistoric life. The drive to ensure the next generation carried costs that the body did not always survive. The cost of reproduction was not limited to the moment of childbirth. It extended across years of repeated pregnancies, breastfeeding, and physical labor required to sustain offspring in demanding environments. In prehistoric hunter-gatherer societies, children were not raised in static households supported by stored food and medical infrastructure. They were carried across long distances, exposed to fluctuating climates, and dependent on continuous adult effort for survival. For women, reproductive years often overlapped with peak workloads, including gathering plant foods, processing materials, maintaining shelters, and caring for multiple children at the same time. When asking why most humans in prehistory never reach 40, It is important to consider how this prolonged reproductive effort accumulated strain over time. Energy allocation within the body follows biological priorities shaped by evolution. During pregnancy, the maternal system diverts calories and micronutrients to support fetal growth. During breastfeeding, additional energy is required to produce milk. In conditions where caloric intake was inconsistent, the body compensated by drawing on stored resources. Repeated cycles of depletion and partial recovery could influence immune strength, bone density, and overall physical resilience. Modern medical research shows that chronic nutritional stress affects long-term health outcomes. In prehistoric contexts, without supplements or consistent dietary variety, such stress likely compounded across years. The archaeological record offers indirect evidence of these cumulative pressures. Skeletal remains sometimes display signs of anemia such as porotic hyperostasis, which can be associated with nutritional deficiency or infection. While interpretation requires caution, such markers suggest that physiological strain was common. Pelvic structure can also reveal patterns of repeated childbirth. Although precise reproductive histories cannot be reconstructed from bones alone, researchers infer from demographic modeling that high fertility was necessary to balance mortality. Each additional pregnancy increased exposure to obstetric complications and infection. Even if many women survived childbirth, the repeated stress could reduce longevity relative to individuals who bore fewer children. The reproductive burden also influenced male mortality patterns. While men did not experience pregnancy, their reproductive success often depended on providing for and protecting the group. Hunting large animals involved close-range encounters with prey capable of inflicting severe injury. Ethnographic observations of contemporary hunter-gatherers show that hunting accidents are not rare and serious injuries can permanently limit mobility. In prehistoric contexts without advanced medical treatment, such injuries could shorten lifespan. The reproductive phase of life therefore coincided with heightened physical risk for both sexes. Though in different forms, this overlap between peak reproductive effort and elevated mortality risk compresses the demographic window in which individuals might accumulate enough years to approach 40, even if the biological potential for longer life existed. The probability of surviving through repeated reproductive cycles in harsh environments remained limited. Over generations, this pattern became embedded in population structure. High fertility sustained numbers, but the physical toll of sustaining that fertility reduced the proportion of individuals who would experience extended post-reproductive decades, returning to the evolutionary origins of Homo sapiens. It becomes clear that our species' life history strategy balances prolonged childhood with significant parental investment. Extended dependency of offspring increases cognitive development and social learning, but it also intensifies parental workload. The benefits of this strategy appear in cultural complexity and cooperative behavior, yet the costs appear in cumulative strain across the reproductive years. In a world shaped by environmental uncertainty and limited medical support, these costs influence survival probabilities. 40 did not represent a programmed endpoint. It represented an age that many did not statistically reach after navigating the compounded demands of reproduction and subsistence. The reproductive system ensured the continuity of the species, but it did so by prioritizing generational turnover over extended individual longevity. Beyond the physical toll of pregnancy and providing for the group, reproduction carried a more subtle evolutionary consequence that shaped lifespan in prehistoric populations. Natural selection measures success not by how long an individual lives, but by whether genes are successfully passed to the next generation. Traits that enhance fertility, early survival, and the ability to raise offspring are strongly favored. Traits that primarily benefit survival decades after reproduction exert weaker influence on genetic transmission. This principle helps explain why extended longevity was not consistently reinforced during most of human evolutionary history. When considering why most humans in prehistory never reached 40, this evolutionary weighting becomes central: the reproductive years. typically spanning from the late teens into the 30s in many traditional contexts, represented the period of greatest biological importance for gene propagation. Once individuals had successfully reproduced and ensured some degree of offspring survival, the selective pressure to maintain strong bodily repair into later decades diminished. This does not imply that older adults lacked value. Anthropological theories such as the grandmother hypothesis propose that post-reproductive individuals can enhance the survival of grandchildren by contributing knowledge, childcare, and food acquisition. Some demographic studies of contemporary small-scale societies support the idea that grandmothers positively influence child survival. Yet even if such contributions offer evolutionary advantages, they operate within a probabilistic framework. If environmental hazards prevent many individuals from consistently reaching advanced age, the selective force preserving late life maintenance remains moderate rather than dominant. The biological systems responsible for cellular repair and immune regulation reflect this balance. Mechanisms that protect against acute threats during youth and early adulthood are strong because failure during these stages directly reduces reproductive success. Mechanisms that prevent gradual degenerative processes over long spans may receive less consistent reinforcement if relatively few individuals historically lived long enough for those processes to influence reproductive output. Modern biomedical research identifies trade offs between early life growth, reproductive investment, and long term maintenance. Rapid growth and high reproductive output can correlate with accelerated aging in some species, while humans exhibit comparatively long lifespans among primates. Our extended longevity likely evolved in the context of cooperative social systems rather than guaranteed survival into advanced age for the majority. In Pleistocene environments characterized by mobility, fluctuating resources, and persistent injury risk, the demographic window for reproduction was limited. Those who survived to reproduce transmitted genes effectively, even if they did not consistently live into their 50s or 60s over many generations. This pattern shaped a species whose biology supports survival through reproductive years under stress, but does not guarantee sustained protection against cumulative wear beyond that phase. The outcome is not a rigid ceiling at 40, instead, it is a probabilistic landscape in which reaching 40 depended on surviving childhood hazards, reproductive risks, and environmental pressures that acted most strongly before and during the primary reproductive period. Archaeological evidence suggests that some individuals did live beyond 40, demonstrating that the human body is capable of extended survival. However, their relative rarity in many prehistoric samples indicates that longevity was unevenly distributed. The evolutionary logic did not require universal long lifespan to ensure species continuity. It required sufficient survival to maintain generational replacement. Understanding this evolutionary weighting clarifies why 40 was less common as a lived reality in prehistory. The biological architecture of our species reflects a long history in which the urgency of reproduction outweighed the expectation of extended old age. Longevity beyond the reproductive peak was possible, sometimes even beneficial, but it was not the primary target of natural selection across most of human evolutionary time. In prehistoric life, physical injury was not an unusual interruption. It was an expected possibility woven into daily activity. Hunting large animals required coordination, courage, and closeness to creatures capable of inflicting lethal harm. Gathering plant foods involved traversing uneven terrain, climbing, digging, and carrying heavy loads. Movement across landscapes shaped by rocks, rivers, forests, and seasonal ice introduced constant risk of falls and fractures. When examining why most humans in prehistory never reached 40, the cumulative effect of injury without advanced medical care becomes a decisive factor. A broken limb today is typically immobilized, surgically stabilized if necessary, and protected from infection. In prehistoric contexts, recovery depended entirely on the body's natural healing capacity and the support of the group. Archaeological evidence reveals numerous healed fractures in ancient skeletal remains, demonstrating that individuals sometimes survived significant trauma. This survival implies social cooperation. Injured members were likely fed, protected, and assisted during recovery, yet not all injuries healed. Compound fractures could become infected. Severe head trauma could prove fatal. Even non-fatal injuries could permanently reduce mobility, limiting an individual's ability to hunt or gather, thereby increasing vulnerability to future hardship. The margin between survival and death remained thin. Skeletal remains from Pleistocene sites across Africa and Eurasia show patterns of trauma consistent with physically demanding lives. Rib fractures, forearm breaks, often interpreted as defensive injuries, and signs of repetitive stress on joints illustrate ongoing exposure to strain. Degenerative joint disease appears in some individuals well before what modern societies classify as old age. Repeated mechanical stress combined with nutritional fluctuation may have accelerated functional decline. In an environment where daily subsistence required strength and mobility, even moderate impairment could compromise survival prospects. Infection magnified these risks. A wound that penetrated skin provided entry for bacteria. Without antiseptics, sutures, or antibiotics, the immune system bore full responsibility for containment. While many infections would have resolved naturally, others could spread rapidly. Sepsis, though invisible in skeletal remains, would have been a real threat. The prehistoric world did not need large-scale epidemics to shorten lives. Localized infection in a small group could claim individuals unpredictably. Environmental exposure also increased injury risk. Seasonal shifts during the Pleistocene created cycles of extreme cold and heat. Cold environments increase the likelihood of frostbite and accidents on ice. Heat stress can impair judgment and endurance. Long-distance travel to follow migrating herds or locate water-exposed groups to unfamiliar hazards. The human body evolved remarkable adaptability, yet adaptation does not eliminate risk. It merely improves odds within constraints. Ethnographic studies of contemporary hunter-gatherer populations provide cautious insight into injury patterns. Rates of accidental trauma remain significant, particularly among adult men engaged in hunting. While direct comparison to deep prehistory must be careful, these observations illustrate how subsistence strategies inherently involve physical danger. In contexts without modern healthcare, injury contributes meaningfully to adult mortality rates, returning to the broader question of why most humans in prehistory never reach 40. Injury operates not as a single dramatic cause, but as a steady background pressure. Over years of active subsistence, small and large traumas accumulate. Some heal imperfectly, leaving lasting impairment. Others result in death. Combined with high childhood mortality and reproductive risks, injury narrows the proportion of individuals who survive long enough to approach middle age. The prehistoric environment did not require catastrophic events to limit lifespan. It required only the persistent presence of physical risk in a world where medical care was absent. Within that world, 40 represented an age reached by some but statistically constrained by the everyday hazards of survival. Injury in prehistoric life did not end with the visible wound. Its deeper impact often unfolded over months or years through chronic pain, reduced mobility, and secondary complications. A fractured limb that healed at an imperfect angle could permanently alter the way someone walked. A damaged joint might stiffen, limiting range of motion. Repeated stress on knees, hips, and shoulders from carrying loads or throwing spears left subtle but lasting marks on the skeleton. When asking why most humans in prehistory never reached 40, it is not only fatal trauma that matters, but also the gradual erosion of physical capacity caused by accumulated strain. Archaeological analysis of ancient skeletal collections reveal patterns of degenerative joint disease in individuals who died in what modern societies would consider early adulthood. Osteoarthritis, characterized by joint surface wear and bone remodeling, appears in many prehistoric remains. While such degeneration does not immediately cause death, it reduces functional efficiency. In a subsistence economy where daily survival depended on physical performance, diminished mobility could compromise access to food and increase dependence on others. Social cooperation likely buffered some of these effects. Yet prolonged impairment would still elevate vulnerability during periods of scarcity or migration. The absence of advanced medical care amplified even minor injuries. Today, a deep cut is cleaned, disinfected, and protected. In prehistoric contexts, wound management relied on natural healing processes and possibly traditional remedies derived from plants. Some ethnographic studies suggest that foraging societies possessed knowledge of medicinal herbs with antimicrobial properties. However, such treatments would not always prevent infection. A wound that healed externally might still harbor deeper complications. Chronic infections can leave subtle skeletal markers, but many would have left no trace detectable thousands of years later. Their presence must be inferred from contextual understanding of pre-modern health conditions. Repetitive microtrauma also contributed to long-term wear. Activities such as digging for tubers, scraping hides, pounding plant fibers, and transporting carcasses imposed repeated stress on muscles and joints. Over years, these movements produce cumulative strain. In industrial societies, occupational overuse injuries are mitigated by rest periods, ergonomic tools, and medical treatment. In prehistoric settings, rest was often limited by necessity. Food acquisition could not pause for long recovery without risking hunger. This dynamic created a cycle in which minor strain compounded, gradually reducing resilience. Climate conditions intensified physical burden. During colder glacial phases, maintaining body temperature required additional energy. Cold environments stiffen joints and increase risk of muscle injury. Icy terrain elevates the likelihood of falls. In warmer regions, dehydration and heat stress impair endurance and coordination. Across diverse ecosystems occupied by Homo sapiens after dispersal from Africa, environmental extremes interacted with physically demanding subsistence tasks to shape daily risk. Researchers studying skeletal stress markers have identified patterns such as enthesopathies, where muscle attachment sites on bone show signs of repetitive strain. These markers reflect heavy sustained physical activity. While such activity also indicates strength and adaptation, it simultaneously reveals that the body operated under continual mechanical load. Over time, sustained load accelerates tissue wear. In a population already shaped by high childhood mortality and reproductive stress, the added burden of chronic physical strain reduces the number of individuals who maintain full functional capacity into later decades. Injury therefore operates on two levels. Acute trauma can abruptly end life. Chronic strain can gradually erode the capacity required for survival. Together they narrow the demographic window in which individuals remain healthy enough to approach 40. The prehistoric body was resilient, yet resilience has thresholds. In environments without surgical repair, physical therapy, or antimicrobial drugs, those thresholds were more easily crossed. Understanding the cumulative nature of injury deepens the explanation for why 40 was statistically less common in prehistory. Longevity depended not only on avoiding catastrophic events, but also on withstanding the slow accumulation of wear across years of unrelenting physical demand. Violence between humans formed another layer of physical risk in prehistoric life, one that is visible in the archaeological record, yet difficult to measure precisely. Small mobile groups competed over territory, water sources, migration routes, and access to game. While cooperation characterized much of human survival, conflict also occurred. Skeletal remains from various prehistoric sites reveal embedded projectile points, cranial fractures consistent with blunt force trauma, and defensive injuries on forearms. These traces suggest that interpersonal violence, whether episodic or recurring, contributed to mortality in some communities. When asking why most humans in prehistory never reached 40, conflict cannot be separated from the broader landscape of risk. Violence in small-scale societies does not require large armies or organized warfare to influence lifespan. Even limited skirmishes between groups can result in fatalities among adults in their physical prime. In populations already shaped by high childhood mortality and reproductive strain, the loss of young adults further narrows the proportion of individuals who might eventually approach middle age. Demographic modeling shows that when mortality concentrates in early and mid-adulthood, average lifespan remains low even if some members survive beyond those years. The evolutionary environment in which Homo sapiens developed included both cooperation within groups and competition between them. This dual dynamic shaped social structures and survival strategies. Archaeological evidence from sites in Africa, Europe, and the Near East indicates episodes of interpersonal violence stretching back tens of thousands of years. For example, skeletal collections from late Pleistocene contexts sometimes show multiple individuals with trauma patterns suggesting conflict, although the scale of violence varied across regions and time periods. The presence of injury from weapons confirms that physical confrontation formed part of prehistoric reality. The consequences of such conflict extended beyond immediate fatalities. Survivors of violent encounters often carried lasting injuries. A fractured skull or damaged limb could impair function long after healing. In the absence of surgical care, complications might arise from infections or internal damage. Even psychological stress, though not visible in bones, likely influenced group cohesion and decision making. Repeated exposure to threat demands vigilance and physical readiness, which may divert energy from long term maintenance processes within the body. Environmental instability sometimes intensified conflict. Climatic changes during the Pleistocene altered resource distribution. Scarcity increases competition. Groups forced into overlapping territories may have faced high tension. While some archaeological periods show evidence of peaceful coexistence and exchange networks, others reveal mass graves or clustered trauma consistent with violent episodes. The variability underscores that conflict was not universal at all times, but it was a recurring possibility within certain contexts. From a demographic perspective, mortality due to violence disproportionately affects young adults, particularly males engaged in defense or hunting. When prime age individuals die, generational overlap decreases. Fewer adults remain to reach later decades. Even if biological aging processes would permit survival beyond 40, the probability of doing so declines when early adult mortality rises. This pattern parallels what demographers observe in modern contexts affected by conflict. Life expectancy drops sharply when violence concentrates among younger cohorts, returning to the central question. Violence operates as another cumulative pressure layered on top of injury, infection, reproductive risk, and childhood mortality. It does not require continuous warfare to influence average lifespan. Intermittent lethal encounters spread across generations are sufficient to shape demographic outcomes. The prehistoric world was not defined solely by harmony or by constant conflict, but by a complex interplay of cooperation and competition. Within that interplay, the possibility of violent death formed part of the environment in which human biology evolved. 40 therefore was not only limited by internal physiological factors but also by external social dynamics that reduced the number of individuals who survived. Long enough to reach that age, long before written records, long before cities or domesticated animals reshaped the microbial landscape. Infectious disease formed a quiet and constant presence in human life. It did not always appear as dramatic epidemic waves. Often it manifested as localized infections, parasitic burdens, gastrointestinal illness, or respiratory disease that moved through small groups and faded only to reappear later. In prehistoric contexts without antibiotics, vaccination, or controlled sanitation, the immune system stood as the primary defense. When considering why most humans in prehistory never reach 40, the steady pressure of infection becomes a central piece of the explanation. Humans evolved in close contact with soil, water, animals, and one another. Pathogens do not require dense urban populations to exist. Many bacteria and parasites thrive in small communities where hygiene is limited to natural water sources and organic materials. Minor skin wounds provided entry points for infection. Contaminated water could transmit intestinal pathogens. Zoonotic diseases transmitted from animals hunted or scavenged introduced additional microbial challenges, while some infectious diseases associated with agriculture and large settlements likely intensified after the Neolithic transition. Pre-agricultural humans were not free from disease. The archaeological record reveals indirect evidence of infectious burden. Certain skeletal lesions suggest chronic infection such as periostatus, which reflects inflammation of bone surfaces often associated with bacterial invasion. Dental abscesses are common in ancient remains, indicating untreated oral infections that could spread systemically. Tuberculosis, one of the oldest known human diseases, leaves characteristic marks on bone in some prehistoric cases. Although not all infections leave skeletal traces, the presence of those that do suggests that microbial threats were embedded in daily life. The immune system itself operates under energetic constraints. Mounting a strong immune response requires significant metabolic resources. Fever increases energy expenditure. Producing antibodies and inflammatory mediators draws on nutritional reserves. In environments where food availability fluctuated, immune defense competed with other biological priorities such as growth and reproduction. A malnourished child or a breastfeeding mother may have faced reduced immunological resilience. This interplay between nutrition and immunity likely influenced survival probabilities at every stage of life. Infections need not be immediately fatal to shorten lifespan. Recurrent illness during childhood can impair growth and weaken organ systems. Chronic parasitic infections drain nutrients over years, subtly reducing strength and stamina. Even survivors of acute disease may carry lasting physiological damage. In modern epidemiology, repeated infections are associated with long-term inflammatory states that contribute to tissue wear. In prehistoric contexts where repeated exposure was common and medical care was absent, such cumulative effects likely reduced the number of individuals who maintained strong health into later decades. Climatic variability further shaped disease patterns. Seasonal rains can increase mosquito populations, facilitating transmission of vector-borne diseases in certain regions. Cold stress may predispose individuals to respiratory infections. Movement across ecological zones exposed groups to unfamiliar pathogens. As Homo sapiens expanded out of Africa into Eurasia, they encountered new microbial environments. Genetic evidence suggests that some immune-related genes underwent selection during these migrations, reflecting adaptation to novel pathogens. Yet adaptation does not eliminate disease. It merely adjusts the balance between host and microbe. Within this evolutionary and ecological framework, infection acted as a steady filter across the lifespan. Many children succumbed to disease before reaching adolescence. Adults faced risks from untreated wounds, respiratory illness, and gastrointestinal disorders. Even when individuals survived individual episodes, repeated infectious stress narrowed the demographic window in which they might reach 40. The prehistoric body evolved mechanisms to combat pathogens effectively enough to sustain reproduction, but not necessarily to guarantee protection across extended decades. The quiet persistence of infection forms another layer in understanding why 40 was less commonly attained in deep prehistory. Infection in prehistoric life was not only a matter of acute episodes that ended quickly in recovery or death. It often unfolded as a cycle of exposure, partial healing, reinfection, and lingering weakness. In small mobile groups, individuals lived in close physical proximity. Food was shared, tools were passed hand to hand, water sources were communal. While these practices strengthened social bonds and cooperation, they also facilitated microbial transmission. When asking why most humans in prehistory never reached 40, it is important to consider how repeated infectious stress gradually eroded resilience over time rather than relying solely on catastrophic disease events. Parasites likely formed a persistent background presence. Intestinal worms transmitted through soil or contaminated food can persist in the body for years, drawing nutrients from their host. Chronic parasitic infection reduces iron levels, weakens immune response, and impairs growth in children. Although parasites rarely leave direct traces in skeletal remains, indirect indicators such as anemia-related bone changes suggest their possible prevalence. In environments where footwear was minimal and sanitation informal, soil-transmitted parasites would have been difficult to avoid. Their long-term impact may not have been dramatic in a single moment, but cumulative over years. Gastrointestinal illness represented another recurring challenge. Water sources in prehistoric landscapes were shared with animals and subject to contamination. Diarrheal disease, particularly dangerous for infants and young children, can lead to dehydration and nutrient loss. Even if a child survived repeated episodes, interrupted nutrient absorption during critical growth windows could weaken physiological systems. The demographic weight of such illnesses is significant in populations with limited access to rehydration therapy or antimicrobial treatment. Diarrheal disease remains a leading cause of childhood mortality even in recent history. Respiratory infections also shaped survival patterns. Exposure to cold air, smoke from indoor fires in enclosed shelters, and closed living quarters could increase susceptibility to pneumonia and other respiratory illnesses. Smoke inhalation from hearths, though central to warmth and cooking, introduces particulate matter into the lungs. Over time, repeated respiratory irritation may compromise lung function, especially in individuals already weakened by nutritional stress. These factors do not guarantee death, but they increase vulnerability when combined with other environmental pressures. The immune system itself reflects evolutionary trade-offs. It must respond vigorously to pathogens, yet excessive inflammation can damage host tissues. In early human environments, acute inflammatory responses were essential for survival against infection. However, chronic low-level inflammation resulting from repeated infections may contribute to gradual tissue wear. Modern biomedical research identifies links between chronic inflammation and age-related diseases. While prehistoric individuals may not have lived long enough for late-life degenerative conditions to dominate mortality, chronic immune activation could still reduce overall physiological resilience across adulthood. Movement across diverse ecological zones during human dispersal further complicated disease exposure. As Homo sapiens expanded into Eurasia, they encountered novel pathogens to which they had limited prior exposure. Genetic studies suggest that some immune-related adaptations arose through interbreeding with archaic hominins such as Neanderthals. These genetic exchanges may have conferred resistance to certain regional pathogens, yet adaptation is partial and ongoing. Encountering unfamiliar microbial communities likely carried mortality risks during migration phases. Taken together, these infectious pressures formed a continuous background against which all other risks operated. A body weakened by parasitic burden may recover more slowly from injury. A child recovering from diarrheal disease may be less resilient to the next infection. A malnourished adult may mount a weaker immune response to respiratory illness. The interaction between infection, nutrition, and physical strain narrows the margin for long-term survival. Within this interconnected web of pressures, reaching 40 required not only avoiding acute fatal disease, but also enduring years of repeated microbial challenge without irreversible decline. The prehistoric immune system was effective enough to sustain reproduction under stress, yet the cumulative toll of infection contributed to the statistical rarity of extended lifespan in many populations. Infectious disease in prehistoric life did not operate in isolation. It interacted continuously with nutrition, physical strain, and social structure, creating a network of vulnerability that shaped survival probabilities across decades. A single episode of illness might be overcome, yet repeated exposure within an energetically constrained environment could tip the balance gradually. When considering why most humans in prehistory never reach 40, it is this cumulative interaction that becomes decisive rather than any single pathogen acting alone. The immune system is metabolically expensive. Fever raises body temperature, accelerating cellular processes and increasing caloric demand. Producing antibodies and inflammatory mediators requires protein and micronutrients such as zinc and iron in environments where food availability fluctuated seasonally. Mounting repeated immune responses competed directly with growth, reproduction, and tissue repair. A child fighting recurrent infection may divert energy from skeletal development. An adult recovering from illness may have fewer reserves to withstand the next physical injury or reproductive stress. The body, shaped by evolutionary trade-offs, prioritizes immediate survival even if long-term maintenance suffers. This interplay becomes clearer when viewed within the ecological context of early Homo sapiens in Africa and later Eurasia. Seasonal cycles determined food abundance. Periods of drought or cold reduced caloric intake. During such times, immune defense operated under nutritional limitation. Even mild infections become more dangerous in malnourished individuals. Anthropological research in recent small-scale societies shows that nutritional stress amplifies susceptibility to disease. While direct measurement in deep prehistory is impossible, the biological principles remain consistent. Infection severity and mortality rise when energy reserves decline. The archaeological record occasionally preserves indirect markers of chronic disease burden. Patterns of growth interruption in teeth suggest repeated physiological stress during childhood. Certain bone lesions indicate long-standing inflammatory processes. Yet many infections leave no skeletal trace, meaning the visible evidence likely underrepresents the true burden. Paleopathologists must infer broader patterns from limited data, but the convergence of skeletal indicators, evolutionary theory, and ethnographic comparison supports the conclusion that infectious stress was persistent. Social structure could both buffer and amplify disease impact. Close cooperation within small groups enabled caregiving during illness. Individuals unable to forage temporarily could be supported by others. Such cooperation likely improved survival beyond what solitary living would permit. At the same time, close proximity increased opportunities for pathogen transmission. Shared sleeping spaces, communal food preparation, and limited waste management facilitated spread. The very sociality that defines Homo sapiens as a cooperative species also sustained microbial circulation within groups. Migration patterns during human dispersal added another dimension. As groups moved into new territories, they encountered unfamiliar microbial ecosystems. Initial exposure to novel pathogens can produce elevated mortality until some degree of immunological adaptation occurs. Genetic evidence suggests that immune-related genes underwent selection during such expansions, reflecting the evolutionary importance of pathogen defense. Yet adaptation is gradual. During transitional phases, infection likely contributed to demographic instability over generations. This layered disease burden reduced the number of individuals who could accumulate enough healthy years to approach 40, even if a person survived childhood infections, avoided fatal injury, and navigated reproductive risks. The cumulative toll of repeated immune activation and nutritional depletion narrowed resilience. 40 was not biologically unreachable. It was statistically constrained by a lifetime of interacting stressors in which infection played a central role within the evolutionary world that shaped our species. Immune competence needed to ensure survival to reproduction. Sustained protection across extended decades was less consistently reinforced. The quiet persistence of disease intertwined with every other ecological pressure forms a crucial part of the explanation for why extended lifespan was less common in prehistoric populations. Food scarcity was not an occasional crisis in prehistoric life. It was a recurring condition woven into seasonal cycles and climatic shifts. In the Pleistocene environments where Homo sapiens evolved, abundance and shortage followed patterns dictated by rainfall, migration of herds, plant fruiting seasons, and glacial fluctuations. When food was plentiful, groups could replenish energy reserves and support growth. When it was scarce, bodies entered conservation mode. To understand why most humans in prehistory never reached 40, it is necessary to examine how repeated episodes of nutritional stress shaped survival across the lifespan. The human body evolved mechanisms to cope with scarcity. Metabolism can slow, fat stores can be mobilized, and appetite regulating hormones adjust to preserve energy. These adaptations increase short term survival during famine. However, energy conservation carries trade offs. Reduced caloric intake affects immune competence, wound healing, and tissue maintenance. In children, chronic undernutrition can stunt growth and impair cognitive development. In adults, prolonged caloric deficit weakens muscle mass and reduces resilience against infection or injury. Survival may be extended temporarily, but cumulative strain increases vulnerability over time. Archaeological indicators suggest that prehistoric populations experienced periodic nutritional stress. Skeletal markers such as enamel hypoplasia reveal interruptions in childhood growth, often associated with famine or illness. Isotopic analysis of bones and teeth indicates dietary variability across seasons, reflecting fluctuating access to different food sources. In some regions, reliance on a narrow range of foods during certain periods may have led to micronutrient deficiencies. Such deficiencies do not always produce immediate mortality, yet they subtly erode physiological resilience. The demographic consequences of scarcity extend beyond individual health. In years of famine, infant and child mortality often rises sharply. Pregnant and breastfeeding women are particularly vulnerable because their nutritional needs are elevated. Reduced maternal nutrition can result in lower birth weight, which is associated with higher infant mortality. Over successive generations, repeated cycles of scarcity can maintain a population structure dominated by younger individuals with fewer surviving into later decades. This pattern reinforces the statistical rarity of reaching 40. Energy limitation also interacts with physical labor demands. For hunter gatherers, acquiring food required expenditure of calories through walking long distances, digging, climbing, and carrying heavy loads. During lean seasons, the energy cost of procurement may approach or exceed intake. Such negative energy balance accelerates physiological wear. Muscle wasting and reduced bone density can occur when caloric deficit persists. In a context without stored surpluses or agricultural buffers, recovery from famine depended entirely on environmental recovery. The evolutionary origins of our species in variable African landscapes conditioned our biology to tolerate intermittent scarcity. Flexibility in diet, cooperative food sharing, and mobility improved survival odds. Yet adaptation to scarcity does not eliminate its costs. Rather, it reflects a balance between enduring short-term deprivation and sustaining long-term health. Natural selection favored individuals capable of surviving lean periods long enough to reproduce. It did not necessarily optimize mechanisms for preserving bodily integrity into advanced age under chronic nutritional fluctuation. As Homo sapiens expanded into colder Eurasian regions, reliance on seasonal hunting intensified. Large game provided substantial calories when available, but hunting success was unpredictable. Climatic downturns could reduce herd populations, intensifying scarcity. In such contexts, nutritional stress compounded existing risks from infection and injury. The cumulative effect of recurring famine episodes narrowed the proportion of individuals who maintained sufficient health to approach 40. Scarcity did not impose a rigid age limit, yet its repeated pressure shaped demographic outcomes across generations. Within a world where food supply fluctuated unpredictably, extended longevity remained statistically uncommon because survival required continual negotiation with the limits of energy availability. Scarcity did not only reduce the amount of food available, it altered the quality and balance of nutrients entering the body. In prehistoric environments, dietary composition shifted with geography and season. Coastal groups relied more heavily on marine resources, inland groups on hunted animals and gathered plants, forest communities on tubers, fruits, and small game. This diversity supported adaptability, yet it also meant that micronutrient intake fluctuated widely. When certain foods were unavailable for extended periods, deficiencies could emerge quietly. To understand why most humans in prehistory never reached 40, the long-term effects of uneven nutrition must be considered alongside more visible threats such as injury or infection. Micronutrients such as iron, iodine, vitamin D, and vitamin B12 play essential roles in immune function, bone integrity, and neurological health. Inconsistent access to these nutrients weakens systemic resilience. For example, iron deficiency contributes to anemia, reducing oxygen transport and endurance. Iodine deficiency affects thyroid function and metabolic regulation. Vitamin D levels fluctuate with sun exposure, which varies by latitude and season. In high latitude environments during glacial phases, limited sunlight combined with reduced dietary diversity may have influenced skeletal health, although skeletal remains do not always preserve clear signatures of specific deficiencies. Patterns of bone fragility and growth disturbance suggest that nutritional stress was common in certain contexts. The energetic cost of brain maintenance also shaped nutritional vulnerability. The human brain consumes a substantial proportion of resting metabolic energy. This evolutionary investment supports cognition, planning, and social coordination, yet it increases baseline energy demand during famine. Preserving brain function may require drawing heavily on body stores, accelerating muscle and fat loss. In children, inadequate caloric intake during critical developmental windows can have lasting effects on growth and immune strength. Even if a child survives undernutrition, compromised development may reduce resistance to later disease. These effects accumulate over decades. Seasonality further intensified nutritional fluctuation. In temperate and cold climates, winter often restricted plant availability and hunting success. Stored foods were limited before the advent of agriculture and preservation technologies. Groups adapted by drying meat or exploiting seasonal abundance, but storage capacity was finite. Extended periods of scarcity could weaken individuals across age groups. Older adults already carrying cumulative wear from injury and infection might have faced disproportionate mortality during such times. Thus, famine episodes acted as demographic filters, reducing the number of individuals who survived into later decades. Archaeological isotope studies reveal shifts in dietary patterns over time, sometimes corresponding with climatic transitions. These findings demonstrate flexibility yet also underscore dependence on environmental stability. When ecosystems changed abruptly, nutritional sources could collapse temporarily. The human capacity for dietary adaptation mitigated some risks, but adaptation requires time. Sudden environmental downturns likely elevated mortality in vulnerable segments of the population. Energy allocation under scarcity also affects reproductive timing. Nutritional stress can delay puberty and reduce fertility. While this may preserve maternal health temporarily, it also compresses reproductive windows. Individuals who survived childhood famine may have entered adulthood with reduced physiological reserves, narrowing the span during which they could withstand subsequent stress. Over generations, repeated cycles of scarcity influence not only survival but also demographic age structure. In populations where food instability is chronic, life expectancy remains constrained. Even if some individuals survive into middle age, the body evolved mechanisms to endure hunger long enough to reproduce, but these mechanisms operate under limits. Each episode of scarcity leaves subtle marks on tissue, immunity, and metabolic balance. Combined with infection, injury, and reproductive strain, uneven nutrition reduces the probability of sustaining health into later decades. 40, therefore, was not an unreachable biological threshold. It was an age fewer individuals attained because cumulative nutritional instability gradually narrowed the window of robust survival. Scarcity did not arrive as a single dramatic famine and then disappear. It often returned in cycles shaped by climate oscillations, animal migration shifts, drought, or cold phases that could last for years. During the Pleistocene, glacial and interglacial transitions altered ecosystems across continents. Grasslands expanded and contracted. Forests retreated and advanced. Herds changed routes. For small mobile human groups dependent on these patterns, ecological instability translated directly into nutritional uncertainty. When asking why most humans in prehistory never reached 40, it becomes necessary to consider how repeated ecological shocks, compressed survival across generations, climatic records from ice cores and sediment layers show that environmental shifts during the late Pleistocene could occur rapidly. Temperature fluctuations altered rainfall patterns, influencing plant productivity and animal populations. In Africa, where Homo sapiens first evolved, periods of aridity fragmented habitats, forcing groups into smaller refuges or pushing them to migrate. Migration itself carries risk. Travel requires energy, exposes individuals to unfamiliar pathogens, and increases conflict potential with neighboring groups. Even when migration succeeds, it represents a response to resource pressure rather than abundance. Repeated displacement strains the body and the social structure simultaneously. Nutritional instability interacts with demographic structure in predictable ways. During prolonged scarcity, birth rates may decline and mortality among infants and older adults may rise. These patterns are observed in historical populations experiencing famine. While prehistoric data cannot provide exact rates, similar biological responses are expected. A body weakened by chronic undernutrition is less able to recover from injury or infection. Older individuals already carrying cumulative physiological wear may be less resilient to caloric deficit. Each ecological downturn thus trims the upper end of the age distribution. Even if some adults survive, the proportion reaching later decades remains limited. Energy scarcity also shapes immune response. When caloric intake falls below metabolic demand, the body prioritizes essential organs such as the brain and heart. Peripheral systems may receive fewer resources. Immune competence declines under sustained malnutrition, increasing susceptibility to opportunistic infections. A mild respiratory illness during abundance might be survived. The same illness during famine could become fatal. These compounding interactions illustrate how scarcity magnifies other risks rather than acting independently. Archaeological evidence sometimes reflects abrupt population declines coinciding with climatic events. Settlement layers thin, tool traditions shift, and evidence of occupation becomes intermittent. While interpretation must remain cautious, these patterns suggest that environmental stress influenced demographic continuity. Groups that endured repeated scarcity may have maintained population size only through high fertility. Yet this strategy reinforces a youthful demographic structure. With many births and many early deaths, average lifespan remains low even when biological potential for longer life exists. From an evolutionary perspective, flexibility in diet, cooperation, and mobility improved resilience against ecological instability. Yet adaptation does not guarantee survival across extended decades for the majority. Natural selection shaped humans to endure fluctuation long enough to reproduce, not to thrive indefinitely under repeated environmental stress. Each cycle of scarcity acted as a filter, narrowing the number of individuals who accumulated sufficient health to approach 40, when placed within the broader network of childhood mortality, reproductive risk, injury, infection, and conflict. Ecological instability reinforces a consistent pattern. 40 was attainable but statistically constrained by a world that repeatedly tested bodily resilience. The prehistoric lifespan was shaped not by a single limiting factor but by the convergence of recurring scarcity and cumulative biological strain. Within that convergence, extended longevity remained the exception rather than the norm. When researchers attempt to understand how long prehistoric humans lived, they cannot rely on written records or census data. Instead, they turn to bones. The human skeleton, though silent, preserves clues about age, stress, disease, and injury. Through careful analysis of skeletal remains, bioarchaeologists reconstruct patterns of mortality and demographic structure. When asking why most humans in prehistory never reached 40, the evidence drawn from burial, Sites and fossil assemblages provides one of the most direct windows into lived reality. Age estimation in skeletal remains relies on biological markers that change predictably across the lifespan. In children and adolescents, the eruption of teeth and the fusion of growth plates in long bones offer relatively precise indicators of age. In adults, estimation becomes more complex. Researchers examine wear patterns on teeth, changes in the pubic symphysis of the pelvis, cranial suture closure, and degenerative alterations in joints. These methods do not yield exact ages but provide ranges that allow demographic modeling across populations. When aggregated across many individuals, patterns emerge. Numerous prehistoric skeletal collections show a high proportion of individuals who died before reaching what modern societies classify as middle age. Many burials contain remains of infants and juveniles, reflecting high early mortality. Among adults, a substantial number fall within estimated age ranges of 20 seconds and 30 seconds. Fewer skeletons consistently display features associated with advanced age. While preservation bias and sampling limitations must be acknowledged, the recurring pattern across diverse sites suggests that reaching 40 was less common than in contemporary populations. Some well-known prehistoric sites illustrate this demographic structure. Upper Paleolithic burial grounds in Europe, for example, often contain individuals whose skeletal features indicate death in early adulthood. In Africa and the Near East, similar patterns appear, though regional variation exists. Importantly, there are also cases of individuals who clearly survived into later decades. Skeletal evidence of extensive joint degeneration, tooth loss with compensatory bone remodeling, and healed injuries demonstrate that some humans lived long enough to accumulate significant physiological wear. These individuals confirm that biological potential for extended life existed, yet their relative frequency within assemblages remains limited. Interpreting skeletal data requires caution. Not all individuals are preserved equally. Infant bones are more fragile and may decompose more readily in certain environments, potentially underrepresenting early mortality in some contexts. Conversely, burial practices vary. Some groups may not have formally buried all members, leading to incomplete demographic samples. Despite these challenges, when multiple independent sites across time and geography show similar age distributions, confidence in broader demographic trends increases. The skeletal record also reveals cumulative stress markers that inform lifespan interpretation. Enamel hypoplasia, healed fractures, arthritic changes, and evidence of chronic infection suggest lives shaped by repeated hardship. These markers do not specify exact age at death, but they contextualize the biological environment in which mortality occurred. When combined with demographic modeling, they support the conclusion that survival into later decades required enduring numerous physiological challenges, returning to the origins of Homo sapiens in fluctuating Pleistocene environments. Skeletal evidence aligns with ecological and evolutionary theory. High childhood mortality, reproductive risk, injury, infection, and scarcity leave demographic signatures. The bones reflect these pressures in aggregated form. 40 was not biologically impossible. It was visible in some skeletons, yet the majority of remains cluster below that threshold. Through the silent testimony of ancient bones, a consistent picture emerges. The age structure of many prehistoric populations was weighted toward youth, with fewer individuals consistently reaching later decades. This empirical foundation strengthens the explanation for why most humans in prehistory did not commonly attain 40 years of age. Estimating age at death from skeletal remains is a careful process shaped by both biological insight and methodological limitation. Adult bones do not carry a precise time stamp. Instead, they display gradual changes that must be interpreted within statistical ranges. Surfaces of joints remodel with use. Teeth wear down from chewing. Cranial sutures slowly fuse over time. These indicators vary between individuals depending on diet, activity level, and genetic factors. When researchers compile large samples, they construct age profiles that approximate how many individuals likely died within specific age brackets. Within many prehistoric assemblages, these profiles consistently show a concentration of deaths below what modern societies define as middle age. This recurring pattern strengthens the argument that reaching 40 was statistically less common in many ancient populations. The pubic symphysis, joint in the pelvis, offers one of the most informative adult age markers. Its surface undergoes predictable morphological changes across adulthood. By comparing archaeological specimens to documented modern reference collections, bioarchaeologists estimate age ranges such as early adulthood, mid-adulthood, and later adulthood. Although the ranges may span several years, patterns across dozens or hundreds of skeletons reveal population-level tendencies. In numerous prehistoric samples, relatively fewer individuals fall into the most advanced age categories compared to younger adult brackets. This distribution aligns with demographic models shaped by high early mortality and cumulative environmental stress. Dental wear provides another important clue. In hunter-gatherer contexts, diets often included coarse particles from unprocessed plant fibers and grit introduced during food preparation. This abrasion accelerates tooth wear. Individuals who lived longer display significant enamel loss and sometimes complete tooth loss, with evidence of bone remodeling indicating survival after teeth were gone. Such cases demonstrate that some individuals did reach older ages. Yet when comparing the frequency of heavily worn dentition across sites, the proportion remains smaller than in modern aging populations. Tooth wear alone cannot determine precise age, but in combination with skeletal indicators, it contributes to broader patterns. Joint degeneration also informs age estimation. Osteoarthritic changes accumulate over time due to mechanical stress. In many prehistoric skeletons, signs of joint wear appear even in individuals classified as mid-adults. This suggests that physically demanding lifestyles accelerated degenerative processes. However, advanced joint degeneration sufficient to classify individuals as elderly appears less frequently. The pattern does not indicate absence of old age, but rather its relative rarity within the preserved record. It is important to acknowledge the inherent uncertainty in skeletal age estimation. Adult age markers become less precise after approximately 40 or 50 years. Biological variability increases with age, making it difficult to distinguish between late middle age and advanced age reliably. Some older individuals may therefore be grouped into broader adult categories. Even accounting for this limitation, demographic reconstructions consistently indicate that a smaller proportion of prehistoric individuals reached ages beyond 40 compared to contemporary populations. The skeletal record must also be interpreted alongside environmental and evolutionary context. If childhood mortality was high and adult life included risks from injury, infection, reproduction, and scarcity, then fewer individuals would survive long enough for advanced age markers to accumulate. The age profiles derived from bones reflect these interacting pressures. They do not represent a strict biological ceiling, but a probabilistic outcome shaped by lived conditions. When multiple sites across Africa, Europe, and parts of Asia converge on similar distributions, confidence increases that these patterns reflect genuine demographic structure rather than isolated anomalies. The bones do not speak in exact numbers, yet their aggregate testimony reinforces the conclusion that 40 was less commonly attained in many prehistoric populations. Through careful measurement and cautious interpretation, skeletal evidence adds empirical weight to the broader explanation of limited longevity in deep human history. Skeletal evidence does not stand alone. It is strengthened when combined with demographic modeling, genetic insight, and comparison to small-scale societies that live in environments somewhat closer to those of prehistory. When multiple lines of evidence converge, a clearer picture emerges of how age was distributed in ancient populations. In asking why most humans in prehistory never reached 40, it is not enough to observe individual skeletons. One must examine patterns across sites. regions, and time periods, and then interpret those patterns within the broader ecological and evolutionary framework that shaped human life. Demographic modeling uses age at death estimates from skeletal assemblages to reconstruct population pyramids. These pyramids often display a wide base of infants and juveniles, narrowing sharply through adolescence and adulthood,